Assalamu alaikum dear students. Now today's topic is regulation of gene expression. Now gene expression is the mechanism at molecular level. Gene expression is the mechanism at the molecular level. By which a gene is able to express itself in the phenotype of an organism. इसका मतलब है जब कोई जीन कोई पॉलीपेप्टाइड बनाता है तब वो इफेक्ट करता है फिनोटाइप को, एंड वी से दैट द जीन हैज बीन एक्सप्रेस्ड. सो it involves the biochemical genetics. तो इसमें बायोकेमिकल जेनेटिक्स इन्वॉल्व होती है. It consists of synthesis of specific RNAs. बहुत सारे RNAs बनाता है, ये भी हो गया जीन एक्सप्रेशन. पॉलीपेप्टाइड्स बनाएगा, वो भी हो गया जीन एक्सप्रेशन. Proteinaceous biochemicals or enzymes. Proteinaceous biochemicals or enzymes. These all processes are collectively called as gene expression. Now it involves the transcription, that is synthesis of RNAs from genes or cistrons. Now it may involve the synthesis of RNAs. It is transcription. It involves the transcription, that is synthesis of RNAs from genes or cistrons. Now certain genes transcribe, you know that certain genes transcribe RNAs, rRNAs, some transcribe tRNAs, some transcribe over small RNAs, other small RNAs. Now these genes also transcribe the mRNAs. Now, which possess the coded information. You know that these mRNAs possess the coded information. These possess the coded information for the synthesis of polypeptides. Now this coded message is carried to ribosomes. This coded message is carried to ribosomes, and with the help of amino acids and tRNAs, and tRNAs, polypeptides are formed. Polypeptides are formed. Polypeptides are formed. Now, later these polypeptides express the cistron or gene. Now, later these polypeptides express the cistron or gene by forming a structural protein, a proteinaceous biochemical, or enzyme. So, we have a DNA strand. This DNA strand is gene or it is cistron. This cistron will transcribe the mRNA. This mRNA goes to ribosomes. These ribosomes are mostly made up of rRNAs. These rRNAs are transcribed also by the genes with the help of tRNAs. Those are also synthesized from the DNA. This undergoes translation. This undergoes translation. When translation occurs, there is formation of polypeptide. There is formation of polypeptide. This polypeptide may express as the, this is expression, structural protein, or it may express as proteinaceous biochemical, or it may express as enzyme. This whole process is called as the gene expression. So the gene expression results in the formation of a polypeptide. Now the regulation of gene expression is the mechanism of switching off and switching on, mechanism of switching off and switching on of the genes depending upon the requirement of the cells and the stage of development. स्टेज ऑफ डेवलपमेंट। पहले जीन एक्सप्रेशन है, फिर उसका रेगुलेशन। तो रेगुलेशन हो गया, ये जीन स्विच ऑफ होगा या स्विच ऑन होगा, ये सिंथेसाइज करेगा ये या नहीं करेगा पॉलीपेप्टाइड ऑन द बेस ऑफ रिक्वायरमेंट्स ऑफ सेल को, ये रिक्वायर है या नहीं है। एंड द स्टेज ऑफ डेवलपमेंट, कि सेल या ऑर्गेनिज्म जो है ये किस स्टेज पे होगा, किस डेवलपमेंटल स्टेज पे होगा। नाउ द simply the control over the functioning of genes, the control over functioning of genes, the control over functioning of genes, functioning of genes is called as regulation of gene expression. The control over the functioning of genes is called regulation of gene expression. Now, this regulation of gene expression may occur at 4 levels. It may occur at 4 levels. Number 1, transcriptional level. Transcriptional level. That is during the formation of mRNA. Transcriptional level, that is during the formation of mRNA. Next one is processing or splicing. Processing or splicing level. Splicing level, you know it. That is regulation of splicing or terminal additions or modifications. That is terminal additions or modifications. Additions or modifications. At next level is during transport of mRNA. Transport of mRNA from nucleus to the cytoplasm. From nucleus to the cytoplasm. And next level is translational level. Translational level. Translational level. Translational level. In prokaryotes, gene expression is regulated by controlling the rate of insertion of transcription. In prokaryotes, the gene expression is regulated by controlling the rate of insertion of transcription. Now you know genes in a cell are expressed to perform a particular function or a set of functions. The metabolic, physiological or environmental conditions regulate the expression of genes. So it is the metabolic, physiological or environmental conditions, those regulate also the expression of genes. And the development and differentiation. The development and differentiation. The development and differentiation of embryo into adult organisms are also a result of the coordinated regulation of expression of several sets of genes. इसका मतलब है जो कोई डेवलपमेंट होता है या डिफरेंशिएशन होती है एम्ब्रियो में, वो सारा कुछ जो है वो एक कोऑर्डिनेशन है, रेगुलेशन ऑफ एक्सप्रेशन ऑफ सेवरल सेट्स ऑफ जीन्स. नाउ इन प्रोकैरियोट्स, in prokaryotes control of the transcriptional rate of incision is the predominant site for control of gene expression. In prokaryotes, control of the transcriptional rate of incision is the predominant site for control of gene expression. इसका मतलब है प्रोकैरियोट्स में ज्यादातर जो रेगुलेशन होती है वो होती है जब इंसेशन होती है ट्रांसक्रिप्शन की. इन अ ट्रांसक्रिप्शनल यूनिट, जो आप जानते हैं, द एक्टिविटी ऑफ आरएनए पॉलीमरेज एट ए गिवन प्रमोटर इज इन टर्न रेगुलेटेड बाय द इंटरेक्शन विथ accessory proteins, which affect its ability to recognize a start sites. इसका मतलब है कुछ प्रोटीन्स बनते हैं उससे पहले जो फंक्शन ऑफ जीन्स को इफेक्ट करते हैं या उनकी एबिलिटी को इफेक्ट करते हैं फॉर द रिकॉग्निशन ऑफ इंसिशन साइट्स. Now, this regulation of gene expression जो है, it can be best explained by, this can be explained by the studying of operon systems. Operon systems in prokaryotes or in E. coli. The gene regulation is best explained by the operon systems that occur in the E. coli. Now these operons are the gene systems which form a complex of genetic material in playing an important role in regulation of genes. An operon is a part of genetic material or DNA which acts as a single regulated unit. So an operon is a part of genetic material. It is a part of genetic material, you know, that is DNA, which acts as a single regulated unit. Single regulated unit having one or more structural genes, an operator gene, a promoter gene, a promoter gene, and a regulator gene. A regulator gene, that is IRR. And there may be a repressor. There may be a repressor or an inducer or co-repressor from outside. This concept of operons was first given by Francis Jacob. Francis Jacob and Jack Monod. In 1961. The operon concept was first given by Jacob and Monod in 1961. Francis Jacob and Jack F. Jacob and J. Monod in 1961. They proposed the concept of transcriptionally regulated system. They proposed the concept of a transcriptionally regulated system where a polycistronic structural gene, that means number of cistrons, where a polycistronic structural gene is regulated by a common promoter and regulatory genes. Such an arrangement they called as the operon. That is, an operon may have now one or more structural genes, an operator gene, a promoter gene, or there may be a regulator gene. Now, this operator, the promoter, and regulator genes, they constitute the regulatory region. Operator, promoter, and regulator genes जो हैं, they constitute the regulatory region. Now, in prokaryotes, they put forth the concept of different operons. For example, there is lac operon, it is lactose operon, there is trip operon, it is tryptophan operon, there is ara operon, this is arabinose operon, there is his operon, this is histidine operon, there is val operon, it is valine operon, now the operons may be of two types, as regulation of gene expression, inducible and repressible.